Let's look into more factors that affect how quickly the liver can clear drug from the body using the well-stirred model. This time we're going to look at intrinsic clearance. In the well-stirred equation, intrinsic clearance is represented by Cl sub I. Intrinsic clearance is dependent on the rate of metabolism of drug molecules and by excretion of drug in the bile. However, before either of these processes can take place, the drug molecule has to first get into the hepatocytes. There are polypeptides on the cell membranes of liver cells called membrane transport proteins. As their name suggests, these substances facilitate movement of drug molecules into and out of the hepatocytes. One group of membrane transport proteins that are found in the liver are called organic anion transporting polypeptides, or OPs. Changes in the activities of these transport proteins can alter how much drug the hepatocyte has available to either metabolize or excrete in the bile. Two major factors that can change OP activity are genetics and drug interactions. Some people are simply born with differing levels of transporter protein activity. There seems to be significant differences in the OP activity in patients of European, African, and Japanese descent. However, there are also significant differences within these populations, so genetic testing of individuals would be needed before making extrapolations based on population statistics. There are some drug-drug interactions that can alter OP activity. In 1997, the FDA approved cerevistatin to treat high cholesterol. While all statins are thought to put patients at risk for a type of muscle damage called rhabdomyolysis, cerevistatin appeared to be particularly dangerous and was withdrawn from the market after dozens of people died due to kidney failure caused by rhabdomyolysis. Some of the people who suffered rhabdomyolysis while on cerevistatin were also taking gemfibrozil, another cholesterol drug. While some of the problems may have been due to cytochrome P450 interactions, it was also discovered that gemfibrozil was a potent inhibitor of the organic anion transporting polypeptide that moves cerevistatin from the blood into the hepatocyte. This may have caused cerevistatin to build up in the blood, putting the patient at increased risk of rhabdomyolysis. It's very difficult to directly measure the activity of drug transporter proteins, but changes in how quickly drug molecules are moved into the hepatocytes can change how quickly they are metabolized and excreted. Thus, the activity of drug transport proteins is a factor that helps determine the overall intrinsic clearance of a drug as part of the well-stirred model.